I can tell you a little bit about a very new area that's emerging that uses DNA as sort of construction material to build little devices on the nanoscale that hopefully will go on to solve some biomedical problems. And I'll tell you how we got there or how the field is getting there. I'll quickly introduce some of you who've never bothered to worry about how your DNA looks like. Just to tell you that it's composed of 4 kinds of bases. The only thing that you need to know is that A pairs with T and A pairs with T and G pairs with C. And so if you have 2 strands that are complementary, what will happen is that they will form a very nice double helix, which is this iconic image of what DNA looks like. And what you'll see is that it looks like a little a thread, right? And in fact, it is a filament. And you can do with this filament exactly what you could do with a piece of wool. And I'll tell you how we can do that. So because you have a strand, which has a certain sequence of A's, T's, G's, and C's, it will recognize only another strand which carries the absolute complement. So all you need to know is just the letters on one string, and you'll immediately be able to predict what the other string is going to look like. So let me show you a new kind of strand. Okay, so I'm just going to approximate that by this kind of a rod or a filament. Okay, and I'm just going to say, let me extend this with another strand which has no pair. Okay, and so if I were to have an artificial set of strands, all of them like this, then I already know that the 2 rods that carry the exact complement are the ones that are going to stick to each other. Okay? So this is nothing but molecular Velcro. So you can do with DNA what you can do with a bunch of matchsticks and Fevicol, or super glue as you call it here, and stick them together and basically make shapes. But this is on the nanoscale. And just to give you an idea of how tiny this is, I want you to take a strand of your hair and mentally magnify it to the size of a Californian redwood tree trunk, which is about 1 meter across, and 2 nanometers, which is just the width of this filament, is roughly the size of an ant on that tree. Okay? So I want you to now start thinking about how you can actually make these devices. This is what the field does. And so there is a UChicago connection here. The founding father of the field is this guy, Ned Seaman, and you should actually talk to him. He's a crazy genius, a fabulous guy to talk to. And he did his undergrad at UChicago. And just, it was just a year and a half ago that I was, that I moved to UChicago. And when I moved, everyone told me, oh, the undergrads here, are the best in the world. They're the best in the country. And I swear, if I had a nickel for every time I heard that statement, I would not need to write for funding for my lab. And then I started teaching. It was amazing. They really are the best. So what Ned has shown really is that it's possible to actually use this fact that you can stick little nanoscale matchsticks made from DNA in space at these very small length scales to give you these wonderful shapes. And how you do it is the following. So if you think of DNA like a piece of wool, okay, you can knit this piece of wool into a sweater or a sock or a hairband. But what the difference is where you make your connections. Okay. So how you do it is you have a very long piece of DNA, and you have shorter pieces of DNA that are called sort of staple strands. And what a staple will do is pinch 2 regions far away from each other in space. So if you have the right set of strands and throw them in solution with this long strand, what it will basically do is knit this long strand by itself into a very specific shape that you can program into a computer. And so what people have done so far is actually make a variety of all sorts of different architectures on a very, very small length scale, all made from DNA. And this was about the time that I started my lab after a postdoc. And I just looked at these magnificent structures and I was wondering, okay, why do we need to make crockery? on the on the nanoscale. And as luck would have it, this field was invented by chemists, and I came from a chemistry department, and I moved to Bangalore, moved back to start my own lab in Bangalore, and I was amongst biologists. I was a solitary chemist, surrounded by a bunch of biologists, and these guys were studying living things, living systems. And one of the things that I found that was common amongst all of them was the fact that they were studying how cells communicated to each other, how cells made decisions. So, for example, an egg, okay, is a single cell, but you know that this single cell is taking decisions, series of decisions, by the fact that if you keep that egg for enough time or under the right conditions, suddenly a chick pops out, right? So this single cell is taking decisions about how to divide, how to create this organism, and when this organism is going to jump out of this egg. And so that's one example of making decisions. Another example is when suddenly a cell in your body decides it's going to become a tumor. Okay, so it suddenly starts dividing like crazy. Third example is a set of neurons in your brain that talk to each other when you puff on a white stick, and then they feel happy. And afterwards, they say, okay, we need to get more of that. Okay, so that's, again, cells talking to each other and taking a decision. Okay, and these decisions are actually made very much in the way that you and I take decisions. Cells receive input from the outside, perform a computation, and then take a decision. And for a cell, these stimuli are in the form of small molecules that diffuse to the cell. Okay? The cell is computing the identity of the small molecule, the direction of approach of the small molecule, and even the concentration of this small molecule, and taking a view of all of these parameters and then performing a computation. And how biologists have understood this is by fusing proteins inside a cell with a certain special protein called green fluorescence protein, which is a little light in a can. So you take this green lantern and stick it onto your protein of interest, and you can watch it float around in a cell. Okay? And by looking at that, you can basically, they've basically sort of come up with heuristic ways of saying, okay, the cell behaves like this. And what green fluorescence protein does is it basically tells you the abundance of your protein of interest and where it's located. For example, if I were to carry a lantern and then go to the office, just because I go to the office, it doesn't mean I'm doing work. I can sit at my table with a massive headache and do nothing, right? So actually what you want is to have a way by which you can look at the amount of work or the activity of your protein of interest, okay? So what you really want is to say not only where your protein is sitting, but if it's super active, let it glow red. If it's sitting and having a migraine, not doing anything, it glows green. And you should have some kind of a graded response that tells you about activity. And this is the technology that my lab has now established using DNA nanotechnology, where people have been sitting and making machines, okay, making shapes. We've been making machines that try to do some work, and that work is to measure the activity of proteins inside cells, to understand how cells function. And so let me tell you a little bit about this technology. So for the function that we want, okay, we want to make a device, right, which has two components. One, it should be able to measure the activity of your protein of interest. So you need to have a measuring module. The second is you need to have a module that targets it. See, the power of green fluorescence protein was the fact that you knew which other protein you glued it on. The power of that green lantern was you knew which protein was carrying the light, right? And so what we need here is to give this a molecular address. You need to know where in the cell you're putting your measuring module. Then you can say something about it, okay, about what's happening. So let me tell you a little bit about the measuring module. Certain sequences of DNA can fold up into space and bind a small molecule. And if you stick on to fluorescent dyes, okay, or fluorescent molecules, then a fundamental principle of binding is that below a certain concentration of this small molecule, in the unbound state, it's going to glow green, and in the bound state, when there's a lot of this small molecule, it will glow red. And for different proportions of red to green, you'll have a very nice graded response, which will tell you the concentration of this small molecule. So why do we want to measure this small molecule? Okay, if I had a car, if I were to just give you an analogy with green fluorescence protein, okay, and let's say I have a car on a road and I take a photograph, okay, I cannot tell whether that car is moving or whether it was the road that was moving or the car is still, right? But if I have a way by which I can measure, I can take a photograph, but I can measure the exhaust fumes that are coming out of the car, okay? Then I have a way to say, okay, it was the car that was moving and it was moving really fast or it was moving slow. And so in a sense, when a protein carries out work, what it does is it also gives out some exhaust, molecular exhaust, okay, which is this star. So if you can sense that molecular exhaust, then you can start looking at the activity of your protein. And so now the question is, okay, what do you want to sense? We want to sense the star. Where do you want to sense it? We want to sense it inside a particular location. So we need to have a way by which you can target this to a particular place. So this is what our lab is sort of well known for, which is that we've figured out ways by which we can put DNA anywhere in a cell that we want. So let's say, the cell actually has several different organelles or compartments which have very specialized reactions that happen, okay? Just like, say, in an office, you have a kitchen, you have the board meeting, the boardroom, you have the floor, the shop floor, and they all have different functions, okay? So the same is the case for a cell, it's compartmentalized. And so let's say I want to position this device exactly in this compartment. So what we've done is we've identified in this compartment something called a shuttle protein. And a shuttle protein has two characteristics. And I want you to think of O'Hare Airport and UChicago shuttles, okay? So, so you have a bunch of UChicago shuttles, okay? Some of them, maybe one will go to the O'Hare Airport, okay? So you have the outermost membrane of a cell, which is the plasma membrane, and you have a small population of the shuttle out there, and a large population on your target organelle of interest, and we want to get to UChicago. Okay. The second property is that I should have an active exchange between these two shuttles, okay, between the population there and the population here. And so now, if I give this device exactly the right ticket, which is your UChicago ID card, okay, then you can basically latch on to the population at the plasma membrane and then get free transport to UChicago, okay? So you're now able to position your measuring module inside the target organelle of interest, okay, and make a measure of what's happening. So some of you would have realized that if you can position something in a particular location inside a cell, you can also use this technology to send these devices to a particular cell in a whole organism, okay? So if I've figured out molecular GPS to get from O'Hare to UChicago, I should be able to get to Chicago from, I don't know, Germany, okay? So maybe we can target specific cells inside a whole organism as well, and this is what we've just been able to do. And so the overall focus of our lab, the overall overarching goal of our lab is to be able to map every single second messenger. I showed you a bunch of second messengers, which are these chemicals that are important for communication between cells, in every single compartment, in every single tissue, and in every single organism that is of interest to biologists. And with this molecular framework, we want to basically throw this open to all biological communities and all medical communities across the world to actually see, screen for drugs which might have biomedical relevance. Today, this has potent applications in the clinical interface and in biomedical, in the biomedical field, which is why I relocated from India. But I'll tell you what these are, but I also want to tell you that when I started, things did not look like this. Today, if I talk like we know what we're doing, I want to say that 8 years ago, we just did not. So what we have now is the capacity to quantitate the activity of proteins in vivo. In vivo basically is the scientist's way of saying inside something that's living. and we can basically look at ion concentrations inside specific subcellular compartments called organelles. And today I want to tell you about one application of this work, which is to quantitate the function of an organelle called the lysosome, that not many people think about, okay? The lysosome is basically the garbage shredder of the cell. All obsolete material that the cell doesn't need is sent to the lysosome, it shreds it up there, and then reuses it. The cell reuses it to make new stuff, okay? And whenever a lysosome goes bad or goes wrong, it results in several diseases. And you can, just like there are 50 ways to leave your lover, there are 70 different ways you can make a lysosome dysfunctional, and each one is a different disease, okay? So, and the molecular beauty that underlies these diseases is the fact that you have just one genetic mutation. One thing is wrong, one protein is wrong, which means that if you can fix that one protein, then you've been able to solve that disease, cure that disease. And there are, so when I was telling my dad, who's actually a pretty well-read person, architect, about lysosomal storage diseases, he was like, what is that? Then I had to say, do you know Tay-Sachs disease? Do you know Niemann-Pick's disease, Pompe's disease? I reeled off a bunch of names. And he was like, yes, I know all of these diseases. Well, they're all due to a dysfunctional lysosome that's not able to break down material, okay? And one of the key things, so just like your body temperature is a number, your blood pressure is a number, and your heartbeat is a number, okay? For the lysosome, the acidity level of the lysosome indicates that the lysosome is functional. If any of these numbers for you, your heartbeat or your blood pressure or your body temperature is off, that number is off, you are not well, okay? So the lysosome also, a number for that is really the acidity level or the pH of the lysosome. And if you can measure the pH of the lysosome, you can tell how badly off, how terribly misfunctional or dysfunctional this lysosome is. And so this is the technology that we've just been able to, sort of realize for the first time that, okay, so this, although this is not the LSD you think, this is the abbreviation of lysosomal storage diseases. We've just been able to measure the pH in lysosomes of diseased organisms, okay, that are carrying specific mutations for each of these diseases. So I'll tell you why this is important and why we want to do this, okay. Even though there are 70 diseases that are called under this umbrella of lysosomal storage disorders, there is not a single small molecule drug for any of them, okay? And these are all diseases that are rare, they're 1 in 5,000. But I want to tell you that many of these diseases are neurodegenerative, okay? Which means that what's very interesting is that many of these small rare diseases connect to a more common disease. So if you are mutated, if you carry one copy of a mutated gene for which causes Gaucher's disease, okay, you have a 10-fold higher chance of getting Parkinson's. If you have both copies of your DNA mutated for that disease, you will die of Parkinson's, okay? So the connection is so tight. And so many of these rare diseases connect to a more common disease. So if you're able to cure this disease with a small molecule drug, that same small molecule drug could now be available to the more common disease, okay? Because thanks to the Orphan Drug Act, which was actually a brilliant thing for the country to have done, many clinical trials actually speed up drugs for these orphan diseases, for these rare diseases. So if one can cure or one can come up with drugs for these kinds of diseases, they would be available sooner on the market. Now, I can't screen for drugs using mice or humans or larger animals, but I can definitely do it with the small worm that I showed you, okay, which is just 1 millimeter, thousands and thousands of them. So if you can label all their lysosomes, and you can just tell that the pH is off, but you know the pH, what should be the correct pH, then you can try a bunch of drugs on them and pick the one where the pH comes to normal. The drug that works for you is the drug that brings your temperature back to normal, right? Or your heartbeat back to normal, right? So that's a similar way that we're going about this. So this technology really helps you to screen for drugs in a very fast way. So let me tell you how we do this. So the pH is nothing but acidity, and this is the concentration of protons. So I told you that we need to have one bit here, which is responsive to this star, and in this case, this is going to be H+, which is your pH, okay? And so we know that you have a single binding event, and therefore we can quantitate pH using a device like this. Now, when I went back to India, I was quite naive 8-9 years ago, and I said that, we do science for the same reason that we do art, because we are human, and so I refuse to be of any value to anybody. I'm going to do only fundamental science. I refuse to be driven by disease and solving disease. Okay, so, and the time I went back, I had a very idiotic and stupid question in my mind, which was, I looked at this very strange structure of DNA. This is also a structure formed from DNA. It's not this iconic image that you have. It's an unusual structure, okay? But it's formed only at acidic conditions. And the idiotic question that I had was, why is this 4-stranded, whereas DNA is 2-stranded? And then we kind of spent a few years, and we understood it, and we said, well, since we understand it, can we manipulate it? Can we use it for something? And since it formed only at acidic conditions, we ultimately went on to make a pH sensor out of this, where we could make a device that in the absence of protons, in low acidity, glowed green, and at high acidity, glowed red, just because these two molecules, these fluorescent molecules, have to be, their distance between each other should change in order to have this change in color, okay? And so then we said, okay, we have a pH sensor, and it senses pH in vitro in a glass test tube. Can it do the same thing inside a live cell? And so, because we know that there are all of these compartments, can we track this? So can we track this shuttle going from O'Hare Airport all the way to UChicago and take intermediate stops? So the way a cell feeds itself is, it uses a process called endocytosis. And so here we have a particular shuttle. We put the ID card, which is a transferrin, which is a particular protein, and we were able to mark those compartments that were only on the pathway of certain shuttles. And we were able to look at endosomes carrying cargo at each station, okay? So these undergo stages of maturation, which is called endosomal maturation, where it goes from the early endosome to the late endosome to the lysosome. And we were able to show for the first time that you could actually watch endosomal maturation inside a cell. And this created a big flutter, because for the first time people had shown that you could use DNA for something which was not what cells use it for. Cells use it to code for instructions. Cell doesn't use it for pH, to measure pH, okay? So this is a very unusual, it's an artificial task performed by a natural substance, naturally occurring substance. So the next thing that we said was, this is great, wonderful. Can we do this inside cells that are present inside a living organism? And at this point, I was joined by a geneticist, Sunaina, who's now in London. And what she showed was that she could target them to some specific cells. So C. elegans has 959 cells, and this went in only to 6 types of cells. All of them were the same cell type, okay? So you've targeted to a specific tissue. And having targeted it to the specific tissue, we were able to show that it went down a very specific pathway in that specific tissue and performed, if you look at device performance, this is device performance inside a glass cuvette in red, and device performance inside a compartment that's present inside a living cell, that's present inside a living organism. And for me, even more than all these pretty pictures, I think this graph, every time I give this talk, I'm blown away by this graph. Because what it tells me is that you have an artificial material made from DNA. This is an artificial device. You put it inside a living organism, and you know that 10,000 things can go wrong. Yet, this device goes very specifically to a specific cell, to a specific pathway, carries out a specific function that you engineered it to carry out with exactly the same efficiency as it does inside a glass cuvette. And for me, this is really remarkable. That means you can program these devices so accurately that it can really perform quantitatively. And so we then went on to show that you could watch endosomal maturation inside a live organism, which was a first. People had been looking at this for 40 years, but without being able to measure that. And so one of the things that we had always asked was, we can build structures with a precision that you could not achieve with any other scaffold. And therefore, we should also be able to access functionality that we cannot with any other scaffold. What characterizes our work is that these devices are very simple. They're structurally minimalist, but we try to see whether they have function in complex environments. And if these structurally simple devices can have complex function in complex environments, then we can gradually build on complexity to actually start doing interesting things. Because of the richness of the structure that we can actually attain with DNA. I'm going to sort of wind up and tell you that right now what our lab is doing is using DNA, which is nature's material. Okay, nature has been synthesizing you, me, my cat, my dog, my donkey with all the same material. Okay, not that I have a donkey, but okay. So it's using the same material, stitching it up in different ways, and is giving you life forms of stunning diversity, okay? And we are just now realizing the power of DNA and making all these different kinds of shapes. So as engineers, okay? But nature is the oldest engineer. And so we're now trying to see if we can use these devices as a sort of powerful lens to visualize new biology, to understand function of organelles and cells, inside us while we are alive. Okay. And so let me just introduce my team. We moved 1.5 years ago from a time zone which is 11.5 hours. It's nearly as far as you can get the other side from Chicago. This work is highly collaborative. It's mostly done by a bunch of students who've now graduated. These guys are now taking it into the realm of biomedical science. One of the reasons why I chose Chicago over all of the other offers that we had was that I felt this was the place that we could actually had the lowest barriers to interdisciplinary science. The medics, the biologists, they're all uber collaborative, not because they need to, but because they want to. The attitude I see there is, let's get it done any way we can, not worrying about anything else. And I'd be happy to take any questions that you may have.
Are you using the nature of DNA, the fact that it pairs up the way that it does, to replicate? I get the impression you build a designer and then you need to multiply it.
So these devices, we don't need to multiply them. For the moment, what we're doing is we're using them as reporters. Okay? So because we're using them as reporters, we can make them outside and then inject them into you, and they will go and tell you what the system's doing. But these devices are then degraded by the cell. You and I have circulating DNA that's going everywhere because of cells going pop, right? They die and they release their DNA. And then that DNA is removed. So there are surveillance mechanisms that remove DNA. But these devices sort of go under the radar, go to that particular cell, carry out its function, and then they are seen by the surveillance mechanism, and then degraded and sent out of the system. That's how we're working. We're not actually using the replicative function of DNA yet, but that's also possible. Yes. Yes.
And mostly it's not worked very well because lots of off-target things, but it would seem to me like you might be able to put some small molecule on one of these that's actually a therapeutic rather than just a measuring thing. Is that?
That
within
is
the realm
a
of
great
possibility?
question. That is a great question. So, and I'm going to give you the following logic, okay? Science has seen many nevers, okay, which have happened. I'm going to say it's one of the reasons why, Chicago called me was that suddenly we have this super accurate delivery device, which can go to places. We're using it currently to report, okay? We're not using it yet to cure, okay? And the reason is, I would always caution that, it's like getting a reporter to a war-torn zone, okay? I can get one guy and nobody will know the difference, yeah, but I'll be able to get information. But to send a battalion of people, the right number, dosage is very, very important. You have to be able to, and the logistics of getting, say 1,000 people there and solving the problem is a different level of difficulty. Not that it cannot be done, but I think now we have a route, it's possible. But it will be a long time before that would be possible, I feel. But yes, it's certainly something we're thinking about, yes.
Yeah, I have two questions. One is, does this going to apply towards the solution on Alzheimer's? And secondly, when would these be commercially available, your work?
Oh, okay. So so let me talk to you about the first. So Alzheimer's. So the reason why I chose lysosomal storage disorders is that there's a specific lysosomal storage disorder called Niemann-Pick. And Niemann-Pick disease connects to Alzheimer's disease in exactly the same way as Gaucher's disease connects to Parkinson's. Sitting on a technology like this, one has to be in an environment where it then becomes commoditizable, where you are no longer necessary for somebody else to use that technology. Everybody should just use it, right? And so all of this work that I've spoken about is patented, is IP protected, and we're looking for people who will, license out the technology. We're still looking for those people. So if you know of anyone who might be interested, just point them my way. Let them know that there's something like this out there. But thank you for that. Yes. Yes. So, yeah. So N2 is a normal worm, okay? So what you're looking at here is the lysosome in a coelomocyte, which is the cell, which is present inside a worm, okay? So what we're just looking at is the ratio of red to green that you see here. We convert it into some kind of a false color image, okay? But if you look at it in real color, sort of this would be little orange and this would be light green. To make this color difference maximum, we've used the entire spectrum and converted a number. But I just want to say that this represents about pH 5.4. This is what a normal, this is sort of your, see, I've got good reflexes. Still. Okay, so this is what a normal, the normal pH acidity would be in a worm. Yes?
You and your colleagues are doing a lot of this research in India.
Yes.
And I'm curious, are there institutional, governmental, financial barriers or difficulties to doing this research in India? Is that why you came to America? Or could you do just as well this work in India?
So,
how would you compare the two environments?
Yes. No, that's a very, very good question. I want to tell you that the issues are in commoditization. The issues are in taking a finding that you have in the lab post getting a patent, okay? Post getting a patent, what do you do to make something go from a lab benchtop available to the clinic, okay? How can we take these technologies and plug them directly into biomedical problems, okay? So the issue also is the scientific ecosystem that's also in India. There are few numbers of people. We don't have the large numbers of the richness and diversity of problems and the depth that is available in this country, okay? There would be 2 people working on zebrafish, or there would be 5 people working on C. elegans, okay? in UChicago, in just UChicago, we have 6 people who work on zebrafish, 7 people who work on C. elegans, okay, which is different from all of India. So you can imagine the difference in, you don't have to tie your devices to an existing problem. You can just look for the best problem, the most outstanding problem, approach those people and say, here's my technology, would you like to see if we can solve this problem? So this, I think, is a unique advantage that the US presents, that you have a richness in diversity. Plus, nowhere else in the world are people so open-minded to new technologies than here. It's sort of it's the breadth that you have, and it's also the mechanisms and machineries that exist here to push your devices into sort of a commercial, the arena, right? The commercial arena.
This is going to be our last question.
Does your work use CRISPR DNA remodeling technology?
Yes. Yeah.
And if not,
Yes.
will that enhance the application in the future?
Yes. So I think CRISPR technology is, so the question was whether we're using CRISPR technology. Let me just quickly tell you what CRISPR technology is. It's the next big thing, where you can actually surgically edit genomes, okay? So I dream of a time when you can surgically edit a genome. But again, there are several issues, right? So it is a tool, and these are also tools. And I think when one is tackling a problem, a scientific problem or a biomedical problem, one has to use a bunch of tools that are all complementary with each other. I think CRISPR is a great way to tell you what is the protein that is involved, okay? But these tools will tell you the contribution of that, So let's say I have 3 proteins that are each involved in this disease, okay? CRISPR will tell you this guy is involved, this guy is involved, this guy is involved, okay? But it's not going to tell you how much is the contribution of each of them. This will tell you A contributes 20%, B is the major problem, contributes 70%, and C contributes only 10%. So I think a combination of all of this technology with CRISPR would certainly be transformative.
Great. Well, thanks to Professor Krishnan for that fascinating talk. I know I'm standing between you and some more snacks and drinks, so I'm just going to make this very short. My name is Anna Nicotera, and I graduated from the college in 2001. As the president of the Alumni Club of Colorado, I'd like to thank each of you for sharing your valuable time with us this evening. It will not come as a surprise to the people sitting in this room that we have a vibrant and very active group of University of Chicago alumni in Colorado. Our Alumni Club is a volunteer-led group that partners with the university to organize events. In the past year, our club hosted cultural events like a private tour at the Denver Art Museum, a gathering at City Park Jazz, and a meetup at the Cherry Blossom Festival. We organized intellectually engaging events like the Harper Lecture Series and a panel discussion among alumni who are leaders in a variety of Colorado industries. We also put together a number of social and outdoor events like Phoenix Fest, tours of local breweries, ice skating in Evergreen, skiing and snowshoeing, and meetups at sport events. As a club, we welcome all Colorado alumni to participate in events, share ideas about how to make the events better, and volunteer with our local programming efforts. I look forward to hearing from you and seeing many of you at future club events. Earlier this evening, Tracy mentioned the 125,000 engagement goal. And just being here this afternoon, you're helping us to meet that goal. I encourage you to let your impact grow by joining the thousands of alumni around the world who are giving to support scholarships and new inquiry, by attending events and leading reunions, and so much more. So again, thank you for being here this evening and for making our university one of the great centers of education, discovery, and impact. And thank you for bringing your curiosity. So now, please return to the reception. You stick around, talk to your fellow alumni and friends, and enjoy the rest of the evening. Thank you.